Memorial Stadium, which is the home to the Longhorns football team. It's been established in 1924 and it holds over 100,000 fans. So if you're a fan of football, make sure to grab your season ticket so you can come down and enjoy the games.
We have the Little Field statue. This is a World War I memorial completed in the year 1933. This sits at the base of the South Mall, which is a nice grass area where students can study and eat between classes. And this whole pavilion overlooks the Texas Capitol on campus.
This is main building on UT campus, also known as UT Tower. It houses the main administrative staff here at UT. It also has a peregrine falcon named Falcon Girl who lives in the tower. At graduation, the year of the graduating class lights up in the windows and a commencement fireworks fire off from the bottom of the tower. It's a special sight.
The drag is the nickname for a stretch of Guadalupe west of campus. On the drag, there are many restaurants and stores accessible to students. Some notable restaurants include K-Bop, a very delicious Korean restaurant, as well as restaurants such as Kava, Chipotle, and Wingstop.
Welcome to the Department of Psychology. This is the CA building, which houses the Department of Psychology. It was built in 2002. One of the interesting parts of the exterior of the building is the soffit, which is the underpinning of the awning. See, it's a beautiful explosion of red, turquoise, and yellow, and it reflects the original Spanish Mediterranean architecture that's present in some of the very first buildings built on campus. Hey guys, welcome to the courtyard at CAA building. It's a beautiful space for sitting and relaxing typically, but right now we actually have an addition being built over here. So it's a 32,700 square foot addition, a centrally located research space for the Institute for Mental Health Research. Welcome to the CA Library. This is where psychology seminars and section seminars occur. So you can see there's a nice lectern up here as well as some screens, a beautiful view of campus. And typically the chairs are all set up here perfectly for seminars, and if we have social gatherings they can be here as well. And there's also a nice balcony up to the 5th floor that you can see up there. As we go out into the hallway, you can see the administrative offices. And I wanted to highlight that the CA building houses the Department of Psychology as well as the Division of Human Development and Family Services. So there are wet labs, the library, which you just saw, computer classrooms, counseling and observation rooms, faculty and admin offices. There's even a preschool.
Engineering Education and Research Center was completed in 2017. It features cafes, student study areas, a library, and research labs. It's also a great place to come and study, and it has beautiful views of campus.
Hi guys, welcome to the Turtle Pond. Turtle Pond was built in 1939. It's home to dozens of turtles from several species, including the snapping turtle. I recommend bringing some lettuce and feeding them. You'll see them accumulate and come say hi to you.
Hi, we're standing in front of the monochrome for Austin, which is commonly called the canoe sculpture. It was built by Nancy Rubin and it holds over 70 aluminum boats. It stands outside of the Norman Hackerman building, which houses a lot of neuroscience teaching labs, as well as an imaging resource center and the nuclear magnetic resonance suite.
We're standing in front of the Blanton Art Museum right here in this white building. We have Ellsworth Kelly, his art entitled This is Austin. The rest of the museum is 180,000 square feet full of art. We have permanent installations as well as temporary exhibits, and all admission is free to faculty, staff, and students of UT.
We're outside of Gregory Gym, which is the main gym here at UT. It was built in 1930 and named after one of the first 13 graduates of UT. It houses a lot of great activities such as racquetball, rock climbing, squash, as well as offers an indoor walking track, a sauna, and a billiards room with a TV.
Okay, so we are about to go into the building. This is the C building. And when you walk in, you can see this beautiful art that is based on the brain, and I especially like this rainbow out of prism projected onto different brain sections. Okay, so I just came out of the elevator. This is the 6th floor of the C building. It's a very special place because there are 3 offices that belong to behavior neuroscience faculty members. This is Dr. Monfils' office, this is Dr. Jones' office, and that's my office. And as you can see, we have a ping pong table here. And of course, we play some ping pong sometimes. And students are welcome to come up here and play ping pong. This belongs to the department. Now I'm going to walk you guys to the lab. Behind this closed door, we have a couple of behavior neuroscience labs. So this first section here belongs to Dr. Dominguez. And then this section here is Dr. Monfils' lab. And then as we walk further down the hallway, you will see that there are a couple of doors down that way. And this is a huge lab space that belongs to the behavior neuroscience area. Dr. Jones has all of her microscopes in here. Dr. Monfils has her wet lab stuff here. I have some of the stuff here, and then it really belongs to all of us. So this is another place where we can all collaborate and work together. We just came out of the C building, and usually I come out of here and then I start walking towards the Arc, which is only a block from here, and you see that building sort of behind trees with a roof. That's where we are going now. So we are inside of Arc now. This is the low floor, and there are a lot of procedure rooms, places where we keep animal cages and stuff. So here's the procedure room, and then this is behind this wall is where we wash cages and keep the cages and all that. So we are going to go up to my lab now. So we are inside of my lab, and a lot of labs in Arc kind of have similar layout. So this is office space for everybody, for grad students. And this whole section is just wet lab. We do tissue work. We do a lot of immunohistochemistry. So this entire section is dedicated for that. And if you come here, here's a microscope dedicated for analyzing tissue, surgical area here, and then this is the behavior room. We do a lot of behavior conditioning, so standard behavior chambers where we can do Pavlovian conditioning or some other conditioning. So we are 1 floor up, and this entire floor hosts labs of Dr. Jones, Dr. Derville, and Dr. Gonzalez Lima. So this is Dr. Jones' lab space starting here, and then halfway down here is Dr. Derville, and then if we go further out, that's Dr. Gonzalez Lima. And the interesting thing about this setup is that it's circular. So you go all the way around. There are more lab space on the other side, and this entire floor is shared by 3 behavior neuroscience faculty members. So that's sort of the typical layout of ARC.
Hi, my name is Juan Dominguez. I'm a professor here at the Department of Psychology. I'm also a member of the behavior neuroscience area. I've been a part of the university since 2008 when I came here from Washington, D.C. And I'm really excited to have the opportunity to welcome you both to UT and also to give you a brief tour of our lab. I'll begin by telling you what it is that we're interested in studying in my lab. We started with experiments on the regulation of sexual behavior. So we were originally interested in understanding how endocrine hormones, or gonadal were involved in the regulation of mating behaviors. Since then, we've expanded on our understanding of hormonal regulation of behaviors into an analysis of how hormones regulate response to drugs of abuse, like cocaine and methamphetamine. And the link is quite clear, in fact. I think because the regulation of drugs of abuse, or the response to drugs of abuse, is largely dependent on activation of the very same systems that are involved with the regulation of naturally rewarding behaviors like sexual behavior. Anyways, so I will give you a quick walk around our lab. As you can see, this is actually our lab is divided into 3 different buildings. We have one room in the animal facilities where we do our behavior experiments. We have a separate room primarily for storage, and then this is our wet lab where we do our chemical analysis and things related to tissue analysis. So this is our bench, and we have a hood over there, and here is where we process our tissue, our brain tissue, which we collect in the other building over in the animal facilities, and we bring to this space so that we can slice the brain and ultimately process it for the presence of a variety of proteins. The brains are also stored in the refrigerator. This space here is the office space for my graduate students, one of whom is recording this for us right now, but they're both here right behind the camera. And this is our room where we do chemical analysis. So the chemicals that we collect from behaving animals, when we perform techniques like microdialysis, we're able to then analyze the collections or the samples rather, so that we can determine changes in levels of neurochemicals as a function of a variety of behaviors. Anyway, so that's a quick tour of our lab. Again, I look forward to, if all works out, the opportunity to meet each of you in person, and welcome and thanks again.
Hi, my name is Tyler Molesky. I am part of the Curly Lab. Right now I'm going to show you the Curly Champagne Wet Lab space. Here we have a cryostat where we can cut brain tissue or other types of tissue for histology or for punches for different RNA analyses. If you go into the wet lab, you can see that in the middle we have our individual workspaces, and then around the edges of the lab is where we have different instruments where we can utilize for different physiological and neuro outputs. So here are just some of the instruments that we use in our lab. Right here we have a plate reader which we can use for ELISAs. We use ELISAs to look at plasma quart levels in mice and rats and humans. Here we have a bioanalyzer that looks at the integrity of our RNA and DNA before we send it to the core to be sequenced for RNA-seq. And then if we just want to look at individual candidate genes, we can use the PCR instrument to do that. That's it.
So this is another one of our wet lab spaces, and this is where we keep the Kingfisher machine. And this machine can extract DNA or RNA from up to 96 samples at a time. And over here we have what's called a flow hood, and this keeps contaminants out and keeps everything inside sterile, so it's good for like cell culturing experiments. These are our -80 freezers, so this is where we keep all of our biological samples long term. We collect brain tissue, liver, spleen, blood, and saliva. So we may take these samples out, bring them into the wet lab to do procedures like histology, in situ, and extraction of DNA or RNA. This is our microscope room. So here we have a fluorescence microscope. We use this to visualize fluorescently tagged RNA or proteins. So here we have the gene CFOS labeled in red in the hippocampus. And then over here we have a laser dissection microscope. So using this pen, you can outline a brain region on this screen here, and then a laser actually cuts it out. That piece of tissue falls into a tube, and then you can do downstream procedures on it, like RNA-seq or DNA extraction from a specific brain region.
Hi, so this is the office space for the Champagne Curly Lab, both for grad students and postdocs. Right now it is covered in boxes because we're all getting ready to come back and work. This is our conference room where we hold lab meetings. We have a TV where we can present data, and we also have whiteboards to do some brainstorming.
This is the Curly and Champagne Lab animal area where we have surgery rooms, which we'll show you in a minute. We also have housing rooms for mice and for rats. And we also have some behavioral testing rooms down the bottom, and we'll show you each of those rooms.
Hi, I'm Maddie. I'm a grad student in the Curly Lab. This is our surgery room. So here we're all set up with our stereotax and microscope to do surgeries. We do chemogenetic surgeries and implantations for electrophysiology. So we have 4 housing rooms for rats and mice, and we can house them in standard cages, or when we're studying mice in large groups, we use these group housing systems that are designed to mimic a natural burrow system. So usually we have nest boxes down here with tubes connecting them, so it's like an underground burrow system. And then up here is supposed to be above ground. And you might have noticed we are in red light. This is because mice are nocturnal, so we house them on a reverse light cycle. So it's white light at night and red light during the day.
This is one of our behavioral testing rooms. At the minute, it's set up to test rats. And as you can see, these are specialist equipment that we use to study the rat's behavior in a lever pressing test. And this is another of our behavioral testing rooms. At the minute, we don't have much going on, but usually we use this to study mouse behavior, and we often do a lot of tracking of the social behavior of mice using these kind of GoPro cameras with automated tracking setup.
Hello, my name is Linda Noble, and I'm a professor here in the Department of Psychology and Neurology. My research interest could probably be best described as being very translational in nature. And this is because we study preclinical models of traumatic brain injuries and spinal cord injuries. Our long-term goal is to develop better therapeutics to treat these very devastating types of injuries. I teach 2 courses. One is at the graduate level on traumatic brain injuries. I also teach a course to freshman students that's entitled Traumatic Brain Injuries from World War I to the NFL.
Hi guys, welcome to Health Discovery Building, the 5th floor. I'm going to show you the common area here. So here at Del Med on the Health Discovery Building, we have a really nice sort of Zen area, which is a nice break area. During non-COVID times, you can see some beautiful sitting area to have lunch. Now let's go up here and see the lunchroom. Here's the lunch area at HDB. So we have coffee makers, microwaves, fridges. We also have a really nice view out to the construction by the State Building, as well as UT main building or the tower, and UT Stadium. Now we're entering the main lab area here on the 5th floor. There are 2 neighborhoods. The first neighborhood is the neighborhood that we're in, and it's still partly filling up. We have 1 lab bench that's dedicated mainly to immunohistochemistry, PCR, as well as Western, but we can do anything on this bench. You can see this beautiful new lab. It just opened 3 years ago. We're also right next to Linda Noble's lab, so Linda's in the Department of Psychology as well as Neurology, and this is her lab right here. And so they have a lot of expertise that's complementary to ours. Let's go see the cell culture room. This is the Cadet Noble shared cell culture room, so you can see there's a biosafety cabinet as well as incubators, and we culture immune cells as well as neurons. Now we'll take you through the area where you look at Westerns and RNA, as well as immunohistochemistry. So this room has various shared equipment, including the thermal cycler for RT-PCR. Here we have a tape station for looking at RNA sequencing, quality of RNA, as well as a nanodrop. And then we have a Western imager for looking at blots and membranes. We also have a couple of cryostats where you can spend a lot of your time in tissue, as we do in our lab.
Hi, I'm Emily. I'm the Cadet Lab manager here. And I just wanted to show you a little bit more of our workspace. So as you guys have already seen, this is our lab bench where we do all of our wet bench work. Right outside, we have our dry area. This is our office space where we do all of our image analysis, or if you have some downtime and you want to update your lab notebook, look at publications, this is all done right here.
Hi guys, I'm Kate. I'm a new rotating student in the Gaudet Lab, and I wanted to show you the conference room, so come on in. This is a great place if you want to have journal club meetings, or maybe you have a speaker coming, or you want to sit down and talk about data. There's a screen here that you can project anything you want to. We've also got some great views here. So, welcome to the conference room.
Welcome to the Health Discovery building where the Orsini Lab is located. Today, aided by my wonderful graduate student, Leah, I'm going to give you a tour of the wet lab and our animal behavioral testing suite. This is the Orsini wet lab where we perform various staining and immunohistochemistry techniques. Welcome to the rat behavioral testing suite in the Orsini lab. There are multiple rooms in the behavioral suite, and in this space is where we test our rats in various operant tasks that measure executive function, including risk-based decision-making, delayed discounting, working memory, and set shifting. We're able to record behavioral output like lever presses and nose pokes, as well as record videos. We have multiple data streams that are coming out of these test sessions. And another component of this behavioral testing suite is a system for drug self-administration. And in this setting, we can train rats to self-administer various drugs of abuse, including fentanyl and cocaine. And here's an example of our lovely stuffed rat self-administering cocaine. So in this part of the behavioral testing suite is where we perform in vivo electrophysiology. And we can actually couple in vivo electrophysiology with optogenetic manipulation of the brain. So we have a computer system that records neural activity that's coupled to our behavioral system. And we can have our rats perform behavioral tasks while we not only record from brain activity, but manipulate brain activity with optogenetics. In a room across from the electrophysiology room, we have a room that's dedicated to optogenetic testing. And in this room, we have more operant chambers in which we can manipulate brain activity with different wavelengths of light through these tethers while rats perform in different operant tasks. And again, like our other systems, we have a computer system that controls and records behavioral output, as well as we can record video to get a fine-grained analysis of behavior and the effects of these manipulations. In a separate room, we have our setup for performing surgical procedures. And in this setup, we have a system for stereotaxic surgery. But also in this space, we perform surgeries such as jugular catheter implantation, as well as gonadic